everyone. Lab 1.5 in genetics lab is going to be all about an introduction to two great genetic model systems. On the left, you'll see the life cycle of C. elegans, which is a roundworm or nematode, and on the right, we see the life cycle of Drosophila, which is a fruit fly. So where we are kind of within Module 1 is pretty much we've wrapped up Module 1. Lab 1.5 is a transition that's going to take us right to Module 2, where we're going to learn more about C. elegans. So this is going to be a face-to-face lab, or at least there's an opportunity to come in face-to-face, and you'll get to view wild type, which means regular or normal or non-mutated strains, as well as an assortment of mutant strains. And we'll be able to look at both the worms, which is the common name we use for C. elegans, and the flies, which is how we refer to Drosophila. At some point, we will also be scheduling an opportunity to come into my research lab. This will be an option if you're interested in seeing some green fluorescent protein transgenic C. elegans. Okay, so what do you need to do to get ready? Make sure you know the plan. So before lab, there's a pre-lab quiz 1.5 due on Blackboard at 1:30 as usual. In addition, the final version of your figure and legend from Module 1 is due at the start of lab, Monday at 2 p.m. See the Lab Reports tab in the Lab Blackboard shell. That's where you'll upload it. Make sure that you apply all the feedback you got from peer editing. And if you have any questions, please email before you turn it in. The options for Lab 1.5 attendance are going to be described on the next slide. And then after lab, regardless of whether you attend face-to-face or not, there is a Lab 1.5 worksheet on Blackboard. It's more of an online worksheet, kind of similar to the style of our lab quizzes, and that will be due Tuesday, October 5th at 2 p.m. for everybody, and that's 24 hours after our lab starts. If you choose the online option, please email me at least 24 hours in advance so I can plan for how many students will be in person. If you choose that online option, there is a PowerPoint file that has video links so you can look at the wild type, worms, and fruit flies, as well as those mutant strains. And you'll need to take notes on that information and use those notes to complete Worksheet 1.5. You are encouraged to meet virtually with your lab group, or you can work on your own to complete Worksheet 1.5, and that's true for everybody. Okay, face-to-face. If you are going to come into the lab to actually use the microscopes and get to look at the worms and flies, you'll follow the plan on the next slide, and that will tell you if you are scheduled to arrive at 2, 3, or 4 p.m. When you get to lab, you'll be using a microscope to look at, again, wild type and mutant strains, both C. elegans and Drosophila. You'll take notes while you're in lab, and paper will be provided, but please bring a pen or a pencil into lab. You may also want to view the online PowerPoint file to help with Worksheet 1.5. But taking notes in either case is going to be very helpful. And again, as I said, you can meet virtually with your lab group later or work by yourself to complete Worksheet 1.5. So looking ahead to 2 weeks out, and I say 2 weeks out because we have fall break, so we'll be taking a break from lab. We will in 2 weeks have Lab Quiz 2.1. And that will correspond to the following sections of the lab manual. The overview and introduction to C. elegans model system. Part 1 is about the Bargman paper, which is a published research article about chemotaxis using C. elegans. And Part 2 is some more information about using bioinformatics and molecular genetics. So the paper by Bargman is already posted on Blackboard if you want to get a head start. And some of the quiz 2.1 questions are from that Bargman paper. And make sure to complete your pre-lab quiz on Blackboard by 1:30, the usual time. And I'll be posting a Blackboard announcement email to remind you of what to do before Lab 2.1. And then lastly, I want to mention that I'll be sending out a poll to our class to talk about whether or not we want to switch our groups up in lab and how things are working so far.